I want to start off by highlighting the accomplishments of this diatoms.org project and really focusing on the incredible value of the connections that we've made and really been formed between people across the country and across the world, but the incredible value of the connections between people and people and people and diatoms. But this project needs funding, and that brings up a big question about what are we going to do next about that. So we have the opportunity to think about where do we want to go with diatoms.org in terms of funding it, and how might we expand it to be able to include other disciplines in novel ways. And I hope not to talk for too long, but to engage all of you in a conversation so we can have a discussion about these topics. Okay, now. So by any measure, diatoms.org is a success. And some of the ways it's been a success are, first off, just the sheer number of pages that we have now. Almost 200 genera pages that have been created, over 1,100 species pages. And the purpose of these pages is to guide practitioners to correct identifications of diatoms. And that's been one of the activities of the project. Oops. Because of that work, diatoms.org now serves as the standard for identification for water quality and assessments. That is, EPA requires the use of this taxonomic resource in contracts. Diatoms.org is also the backbone for taxonomic certification of analysts through the Society for Freshwater Science. Diatoms.org, of course, hosts the Diatom Web Academy, connecting people through these live seminars and fostering the exchange of ideas and furthering collaboration. And since we started in 2020, actually started by Sylvia Lee as a way to train people through the Taxonomic Certification Committee, our recorded seminars have received over 25,000 views on our YouTube channel. And that's huge. It's a huge reach that we've been able to make. So Diatom Web Academy, where you are now, links us together. Artists, students, academics, teachers, technicians from around the world. Diatom attracts you. And some of the 70+ titles you can read from this list that we've covered a huge range of topics and addressed many different ways that many people think about diatoms and how they use them in their work. Okay. We also feature news on publications, on meetings, on projects, on works of artists such as Stacy Levy. Stacy collaborated with a research ecologist, Judy Lakini in NOAA, to represent giant versions of diatoms that were living in the East River of New York City. So she looked at the diatoms of what's living in the East River and created those on pavers embossed in concrete so that people walking along the promenade can start to relate to these organisms. Oops. Diatoms.org is becoming a source of data in itself. The work of Hu et al. Last year used images from the site. Basically, they scraped every image from the site to be able to use them in machine learning for recognition and classification of images for applications in assessment, ecological monitoring, and biodiversity. Supporting all that is diatoms.org's posts really strong web analytics. In the past year, we had 116,000 active users. Most of those users were actually people that we are called new. And a lot of these people are, say, Googling, what are diatoms? Showing that we have an incredible reach to the general public, to people who didn't know what diatoms are at all. And then we have a smaller number, but 3,000 people are return users, are coming back to the pages, studying the pages, engaging with the science, and those represent 270,000 sessions and 2 million page views in the last year. So it's astounding that we have so much support in terms of the use of this site. Okay. And just to see some of those numbers, so the number of views is something over 1 million. All the page views are 2.4 million, and where those are going, people are going to the genera pages. They're searching directly for something. It could be a Google search. Number 3, diatoms of North America. They're coming in to the main page. What are diatoms? They're Googling on their phone. So we can look at how pages are used by who and how long they stay on those pages, which is a really important measure of how people are taking in that information. And this isn't like, sort of a commercial site where people bounce around. People are in here working, and we can see it in the analytics. But one of the things that I think is the most important, and I'm going to spend some time on it, is that diatoms.org connects human and organism. We are joined visually, emotionally, and scientifically by this resource. Earlier this year, some graduate students from Aalto University in Helsinki, Finland, contacted me and Tim Gasparek, who's the lead designer for this project. So these students kind of stumbled upon diatoms.org, and they were intrigued and inspired, like many of us, by these organisms. And they were also concerned about the levels of pollution in the Baltic Sea. And so they took on a project to see, what if we think about how we can bring these single-celled organisms, can we find a way to have an intimate understanding of the plankton so that people will care about the Baltic and care about the condition of the Baltic. And you can, it looks like it's gotten cut off in the bottom. There's a news piece on diatoms.org where you can see about their work and watch a short film about how they developed an exhibit to bring human and diatom together on an equal scale. And I just think it's brilliant. Here I think this is like marine snow of material just settling down through the ocean. So, and for me, it kind of pulled together the what's sort of magical about this site, that we're able to cross these kind of boundaries and be able to connect from the science that people need to make correct identifications, to inspiring people that have nothing to do with science. But despite all these successes, the project only has enough funding for web maintenance through next year, and that's it. So at the same time, we have this success, but we're out of funding, and now we have these silent extinction, both of species and of taxonomists. Us, the people that are working on these. Okay, I'm getting a message. Oh, great! Thanks for setting out those links, David. That this is a problem happening over and over again across every different group of organism. For us, the Husted collection, holding so many of the important types that were archived there, has been closed for several years. It is being moved from the Alfred Wegener Institute, but it's been inaccessible for several years. This year, Duke Herbarium, who had a major collection of terrestrial plants, announced their plans for closing. California Academy of Sciences this year announced deep budget cuts, losing curators and staff members. And there are talks and plans to drop work, the more kind of morphological work on diatoms for assessment, to be replaced by genomics, because of its being proposed as being faster and less expensive. So, yet at the same time, because of these successes, we're poised right on the edge to forge new approaches in linking data sets and expertise with emerging technologies in genomics and artificial intelligence. Like, what if we think big about what this project can be? And like many organisms, like say bacteria, there's nothing compelling about visuals there. Diatoms are visually, emotionally, and scientifically compelling. Okay. So some of the things I'd just like to run by you folks, after the North American Diatom Symposium that we had in Iowa, I have been having conversation with Jane Marks about submitting an NSF proposal for a research collaboration network with diatoms.org as a hub. Dave, if you want to post that survey, or maybe that you did that, there's a survey. And if you're interested in something like that, you can let us know and give us your email. What can we do linking morphology and genomics? What could we do if we got people in a room to translate across different disciplines? How can we further this power of connection and of organism and human? Besides us, we're all organisms. How can we guide AI to work for analysis in a way that it keeps the unique understanding that we're able to give with our human minds? How can we host and serve large vouchers in new ways? Some of these voucher floras have over 20,000 geolocated images of specimens on slides in permanent archives that have been identified by experienced analysts. I'm going to hazard that there are no other organismal groups that can stand up to that kind of identification that is backed up in a very solid way. So just by the action of making this website, we have gotten a long way. So we want to know your ideas. Where do you want diatoms to go from here? If you go to this link, you can fill out a survey about how you use the site and react to some of the ideas that I put in there, and put in what you're thinking about. Where would you like it to go? And we're running a fundraising campaign to keep the servers in operation. So donate to Dona. You go to this link. Just think, if we have like 2.8 million page views and everybody gave $1 for a page view, we would be well set to be able to, really well funded. So please help us keep our servers open, and let's try to prioritize, let's try to get enough money that we can try to do some of the things that are important to all of you. Okay, the link that you're going to get, or that you can go to from this QR code, I've been wrestling with it for most of this year, and I wish it was better, but here's what you got to do. You go to this link, and if you put diatom in this box, Diatom Lab Research Fund will come up, and that fund is going to go into our account to support diatoms.org. So this is a tax deductible donation through the University of Colorado Foundation. And let's see. So yeah, through this project, we've got an incredible opportunity to connect with each other across cultures and disciplines. And where might we go next? I want to hear from, I can advance the slides, all of you. So, but first I'd like to thank the innumerable minds that shaped this project. That initial support we had from EPA, US Geological Survey, and University of Colorado, from Tim Gasparic as our designer who puts the pieces together, and SoulSpace, our web developers who implement the vision for this site. I'd like to thank the editorial review board, and thanks to all of you who showed up today. I'd like to thank the contributors of Taxon pages, all more than 200 of you. And thanks to the millions of people who Googled, what are diatoms? I think that we've put together something remarkable. So thank you. So David and Hira, I'll stop sharing, and we can open it up for a discussion with everyone. I assume that I'm not getting broken up because I haven't got any messages. So everything okay? Did you just hear that? Okay, thanks. Appreciate it.
Yeah, I think I was gonna let Mark field the questions, but if anybody has any, go ahead and jump in there. There you go, Mark, take it away.
Hey, thanks. Sarah, we did get one that's sort of probably up front in everyone's mind, which is Anna asked us just how much does it cost to keep the servers up for a year? And we're just thinking about absolute maintenance cost of this incredible resource.
Right. Yeah, so I'm including in that cost, I'm about $5,000 a year. So it includes the hosting on the servers, the software fees that we pay that run the back end of it, having the developers fix things that they come up. We have glitches every once in a while, and they're sort of responding to whenever there's something that's not working and doing a fix. So $5,000 a year pretty much covers that base level. And right now we've got that in place for 2025. But I would like to have 5 years in the bank so that we have some security in terms of the web hosting. And I'd like to think about all the other things we need to do. At this point, no one besides the web developers is getting paid for this part. It's a labor of love and community service. So, and I think this is an incredible way for students to be trained, involved in the field. Okay, I'll shut up because that wasn't, you asked what question you asked.
Thanks, Sarah. Yeah, I think it would be really interesting to hear from some other folks of how they use DONA and how they might envision some ways to leverage it. I know for us at my shop, we've used it pretty extensively in a National Park Service project and have basically asked our Park Service program officer to provide us support to develop web pages or develop species pages for the thing. And that's been really
Awesome.
Successful. And
Awesome.
But I'm really interested to hear from others of how you use the page. Is it just checking your taxonomy, or is it other really cool ways that help us broaden our impact in other communities? Don't be shy. Raise your little hand or just turn on your mic.
I think I can just say a little bit. Like here at Retron, we do use it pretty much every day. So we use DONA a lot, and it's like the go-to resource for us, especially if we need to do the text IDs. Also like going for verifying some nomenclature, or also looking at some synonyms and any updates that's usually on the web page too. So it's like Marcus said, we do use it more broadly and mostly in terms of our own taxonomy and ID verification. Maybe other people use it like in outreach, I am assuming, for education that they can discuss or like talk about from their perspective.
How about you, Tom? How are you using it?
Yes, we're using it. Well, we've been fortunate enough to receive new grant funding, and part of that in the proposal we put in that we would all the new taxa, but also taxa that we work with will be making pages for. So genus and species pages that went into our grant, and I certainly encourage everyone else here to do that also.
Hey, Tom, what just out of if it's open information, what funding source was that you actually requested that type of funding for?
That was the NSF. So
was it? Okay, cool.
Yeah, National
Awesome.
Science Foundation. I can't say we requested specific funding. It was part of the part of what we were selling to them. We saying, hey, we were gonna generate these things, and the work on this project was gonna be accessible through diatoms.org.
I just posted project page for Tom's
Yes.
NSF proposal. And as you create taxon pages, Tom, we can link those in through that project. So you see all the things that will be connected to your work.
Cool. Evelyn, I saw your hand go up. Why don't you unmute yourself and turn on your camera and say hi?
Yeah, hi. This is
Hi,
so great to see you guys again and to think about the future of diatoms.org. And our lab uses it in a similar way as I'm sure lots of us, and trying to identify our species and check names and their synonyms and their histories and find references. And I just really love what Tom has done with this grant. And I wonder if there are ways of making that more of a thing, that that when we do go for funding for work that would include some taxonomic components to it, that there's sort of a maybe we would have an idea of how much we should set aside for different sorts of ways that we use the website, as well as time for people to contribute new species. I mean, I know that the people working on new species pages and my group have really learned a lot in doing that, just the process of doing that. And it's helped us understand some of those species better. And to work that process into the funding, that's paying for some results of that. It just seems like something we could have on our minds a little bit more now than ever.
Yeah, and especially if there's ways that we can, have an exchange too. Like what does NSF should eat this up in terms of broader impacts, what you can show by this site.
Right.
What is it that would also help to be able to further in your grant proposal that this project would do to support the work of that?
Yeah, it was when you brought up what, the broader impacts and, making NSF happy or whatever. Now they are requiring that your collections be made available. And any collections that you hang on to need to be made available. And one way of doing that, they've said, is through image databases. And so if you could claim that you're meeting that requirement by contributing species to DONA, or enhancing DONA in some way that makes that material available publicly, I really think that could achieve, then NSF might be more willing to pay for it, because they've already made that kind of a mandate. So.
Yeah, and it shouldn't, it's not, I don't think this is a big ask on NSF. It doesn't have to be a huge amount of money. A small amount of money goes a really long way with this project. One of the things that I thought was really successful was when we had funding from EPA and US Geological Survey to hold workshops in Philadelphia, where we could bring people together and work on a genus in the academy with Marina and her staff, and bring students to be able to learn that process of working in a herbarium and working side by side. And it was a really productive way, just paying people's travel to create some pages with type material in the images.
Yeah, I completely agree with that statement, Sarah. I participated in a number of those trips to the academy, and the amount of work that can get done and just the community getting together to solve problems is just amazing. Hey, Matt, go ahead and turn your camera on and turn your mic on. Let's see what's going on in Texas.
Oh, not a hell of a lot. Nothing good anyway. But I just sort of wanted to build on this idea on using diatoms.org as a mechanism for outreach for grants. Tom already talked about the epizoic grant, and I went hunting for our project page that he put up, and I realized I'd never seen the project pages before. They're hidden under this About Us tab rather than being front and center, next to practitioners and morphology and genera and the glossary. And I think this is a really good way to sort of show the impact of diatoms on these other projects. I mean, I'm looking at the Great Lakes Inventory and Monitoring project page here, where you've got, the relevant taxa, you've got a funding citation, you've got a little project description, you've got links, you've got the participants. This is all really, really useful information that, I think could be front and center a little more.
Okay, that's a good one. I'm putting it in.
Yeah, thanks, Matt. Tommy, turn on your camera and your mic and say hello.
Hey y'all.
Hey, Tommy.
Yeah, I'm counting diatoms right now and I'm listening in. And the most recent comments, I wanted to kind of echo what Evelyn said and what I've been hearing. I had a conversation with Corinna Grease about a year or two ago, who was leading EDI, database management, and talking about how to maintain sustainable funding for that. And something she said was that she really was hoping people would start to put things into their grants for support for data management and databases in the same way that you would write into a grant for overhead, because data infrastructure is a critical infrastructure for everything that we do, and it should be written in as overhead in our grants. And so I was wondering if maybe there's an opportunity for DONA to kind of leverage a similar thing, because DONA is a data repository of sorts. It's an intellectual, crowdsourced information repository, visual based nonetheless, but I think that we could make a case that that is the sort of thing that deserves merit for those sorts of funds to be applied for support.
Yeah, thank you for that. And remember that one of the rules that we have for this project is every image in the genus and species pages on that lineup needs to be on a slide in a herbarium. And while there are many, in biodiversity inventories are really skyrocketing really around the globe, that people are seeing things. One of the things is those inventories are not backed up by specimens, or they're simply images. And so it becomes hard to rely on them in the way that these are images that have a very they're geolocated, they're in collections. People can go back to those very things. There's often chemical data associated with them. So this is like a really powerful amount of information that we have. And in pointing out those voucher floras, those are the same thing. And that's kind of why I'm bringing those up, is we have these tens of thousands of images that we haven't harnessed that could potentially be ways that we could fortify geographic distributions. Is there a way that we can import them into the site? Can we use them in spinning up on the spot floras based on where people are actually working? I would be so excited to just get in a workshop with everybody and figure it out, like, how can we do that? And get technical people in of what can we do that just leverage it so we're able to do it in a scripted sort of way.
That sounds great. We've had some really nice comments in the chat that just resonate how important DONA is for everyone. Anna said, you know, the taxonomy is invaluable on its own, but I often read the ecology and distribution parts of the pages. And I just want to echo that. That's where I love to start to figure out where these diatoms are supposed to be or where they've been seen before. And some things it just provides sanity checks during our IDing process. Heather said at Larrett, DONA is the first go-to for diatom ID. We do all the algal groups as a way to identify lake and reservoir stress. We'll be donating. Thank you, Heather. Really appreciate that. And thanks. And again, says thanks for all that great work that goes into DONA. And Addison from Indiana, Department of Environmental Management, says we're always using it for checking and reading up on ecology and especially tracking name changes. And they're very grateful for this this resource. And Layla, checking in from Paris every couple weeks for our Web Academy, brings that international impact for this thing, not just for not just for our Web Academies, but also the fact that everyone checks DONA and compares the taxonomy of stuff in Europe to the taxonomy of stuff in America. And so it's really an important international resource, not just a North American resource. And that impact is really well heard out there.
I'll add too that I've been in contact with Mike Gowrie from AlgaeBase, who contacted me about some orthographic changes, meaning names that there's something about the spelling which should be corrected. So I've been working through those changes, and you may see that there are a lot of updates going up with some little letters changed around. But now many of these are now linked within AlgaeBase. And one of the next things I'll do with the developers, this is a small thing, so it's going to be covered in what we can do, is to change the way we present the names so it's easier for Mike to connect to diatoms.org through AlgaeBase so that his pages will talk to each other. So we've got yet another check on taxonomy. And then if anybody doesn't know this, you can go and download the entire text, the names and the changes, any kind of updates through the species page. It says download data, and you can get the entire thing and see all the synonyms and changes that may have taken place over time.
I'm glad you bring that up, Sarah, because as I'm thinking of, how we use the website, and I think all of us, our primary reason is to figure out what this thing is we're looking at on the microscope, but what are some of the other things that I use diatoms.org for? One, honestly, is checking my spelling when I'm putting together some figures and I want to label the diatoms on a stratograph or whatever. I'll just go double check my spelling because I know I make mistakes. And one other area that I think is often hidden to folks is the glossary, and I think it's a really, really huge resource. And when I start to think about checking names and looking for genus descriptions and using the glossaries, I really start to think about how, in a way, the diatoms.org is replacing the Diatoms book in my life as far as a go-to resource.
And thanks to Rob Kimmick for that, who's our glossary editor for adding images and references and backing things up.
Hey Sarah, hey Mark. I just wanted to chime in from my own experiences, kind of echoing what David said. I've also used the glossary, especially helping me when I'm learning the different parts of the diatoms. And I not only use it for all the other reasons that everyone else said, but I also use it to educate others around me. I know I talk to a lot of people about what I do, and they get very excited, but very confused. And so taking them to diatoms.org is like the first place that I direct people, my family members and all my friends. And that's like a space where they're able to get really excited because of the beautiful images that are available there. So that's like a resource that I'm always pointing people to, and it helps them connect with me as a scientist, and it helps them feel like they can understand what diatoms are. And I've also used diatoms.org for an outreach program at our school. We're working on in my department. They're putting together like this K-12 outreach event where they're having different labs at FIU put together different little showcases of what they study. And so we use a lot of the great information about what are diatoms and some of the really cool facts in a little poster that we put together that we're able to share with young students and show them pieces of microbial mats under the microscope. And so we've used that in like early education environments as well. And the other way that I use diatoms of North America is just for creating my own art. I go into these beautiful images and I base what I draw and paint off of what I see. And so I'm not only using it for my own identification purposes, but also to create the art, the beautiful diatom art that also helps communicate the importance of diatoms to the broader community.
That's an awesome page. Thank you. And if you would ever like to post on a news page, whether it's the outreach you do, pictures of, just tell us the story of who are you reaching and what are they doing? Because it feeds back into sharing and and with your artwork as well. I think that's really what one of the things we can do because it just amplifies the power of the resource. Thanks so
Yes,
much.
of course. I would love to do that sometime. Thanks for telling me about it.
I just want to follow up real quick, Paige. I got a text on Friday night from an old friend who said they were sitting at the bar with a bunch of people and the discussion of diatoms somehow came up and they said, "Oh, Dave, you can tell us about that." And so sure, I told them about the oxygen production and being part of the food web, but then I sent them to diatoms.org and maybe 30 minutes later, I got this message like, "Oh, we've been scrolling through and looking at all these pictures and they look so cool." And it's a really great way to show people what diatoms are.
That's so amazing. That's so cool that you were able to share. I feel like that's the easiest way to just get people connected. And there's only so much that I can say, and the pictures always speak 1,000 words.
I agree. Hi, Laura. Go ahead and turn on your mic and say hello.
Hello, everyone. So just to go off of what both Paige and David were saying, I use Jonah a lot with outreach stuff at the Academy because it's a great opportunity to kind of have a ton of pictures and to show the morphological diversity of diatoms. And I was actually sitting with one of our marketing personnels who help write blog posts for the Academy. And they were wanting to do kind of an introductory on how to get into diatoms, like ways to process slides at home, safe ways to become like a diatom enthusiast if you don't have a lab. I looked at Dona and there's a lot of great resources, but I was actually wondering if there could be kind of like a beginner's guide to diatom or like even a way you could search it that would be a lot more user-friendly to people who've never seen diatoms before or just even learning about the basic morphological differences between them.
Yeah, that's a good one. Yeah, sort of thinking about different levels of entry for people into the resource, wherever they are at, a way for them to interact with it directly.
Yeah, it can even be maybe like for schools, they could use that as a tool to start introducing high school students or even middle school students, make it like age appropriate. Even elementary kids, there could probably see something, an activity or a game they could play that would be interesting to add like an addition to Dona.
Yeah, and that part is in what our diatoms part is kind of the public part. where there are individual pages, but perhaps that would be something that could be filled out on different topics.
I like that idea.
Please put these in too. I'm writing some things down, but I, urge you to take the survey and add them there as well, so we can record everything that people are sharing. And how about this? Are there any features or processes that any of you would help you do your work? Either make it more streamlined or more, or confirm the work, your IDs in a better way, or is there anything that I see people like you, Mark, looking up into the sky?
Just thinking.
Yeah, look up into the sky. What would
I
that
have,
be?
I have one, Sarah.
Okay.
So actually most of my work now is focused on cyanobacteria. Most of the projects are, but I do the full phytoplankton counts, obviously, including the diatoms. So it would really be helpful for me if there were images of live specimens to go along with all the SEM images.
Yes, images of live specimens. And how are you using that? What in your, this is in what's kind of, is this your routine analysis, or is this outreach, or what's the context of
For the quantitative phytoplankton counts, which are part of pretty much every project I'm a part of. So yeah, for research purposes. Got
it. Do you take live images when you're doing your work? Can you also?
Yeah, I do.
Us?
Yeah, I would love to contribute. It's just I'm so swamped. I need to get a lab assistant, but I have a ton of images, and I would love to sit down with some of the samples that I have that have like really cool diatom assemblages in them, and then just sit down and take like really pretty high resolution images of those. And I can do them. They're, preserved samples. It's just not something I have the time to do right now, but yeah, I don't know, maybe we can come up with something and get a little funding on the side where that could be a little project that I could use existing samples to get some live images of either stuff we already have ID'd on the site, or, as part of new species pages.
Right. And Tom Frankovich and the Marine Dream Team have been leading that in the marine species of putting in live images, and they're fantastic to be able to see the colony structure and the chloroplast structure. I mean, they're really spectacular, and it's a great addition if people are working with live material to take those images, and we can add them into existing. We have a place to put them.
Oh, cool. So Tom, you already have uploaded some live images to the website? I probably just haven't found those species because they're marine and I mostly work with freshwater stuff. So that's probably why I haven't seen them.
Oh, you're on mute. Yourself, Tom.
Sorry, yeah, yes, those are marine, and after this meeting, I will send you a voucher, Flora, that has live images of the freshwater plankton, Lake Okeechobee area, so.
That would be amazing, thanks.
I have a question for Sarah, if I could. Well, first of all, thank you for bringing, all of this to our attention, and trying to keep diatoms.org going well into the future. Is there any way that you can or keep track of how many donations, how much money have we received so far? Is it?
Oh, well, I just opened this today, so I don't know.
Okay, but you have a way of also looking at something at the end of the day, say how much was brought in today? Is there a way for that?
I don't know. I don't have it directly. I need to contact the university to be able to check with them, but I'll go ahead and do it, you know.
Because
and
I would like to,
what the process is.
sure. I would like to kickstart this and maybe, pose a challenge. If, our viewers here today, I will match today's donations up to $500. So if that inspires any of you to contribute and see if we can get somewhere towards, funding our first year of, web support. So if you can keep track of that, Sarah, I promise you can hold me to it and see what happens.
That's fantastic, Tom. That is fantastic.
Awesome.
Thanks so much for inspiring the charge. Really appreciate it. That's great.
That's great.
Okay.
I just wanted to say, Rob brought up something a bit ago in the chat, Sarah. I think one thing that Rob's been, as we know, has been really working hard on the glossary section, but Rob brings up a thing that, if you see something that is wrong on DONA or needs to be fixed on DONA or could be helped on DONA, please, reach out with your skill set as well as making sure everyone's aware of the issues that you're seeing, so that we don't perpetuate things like that, or we make it better. So thanks for bringing that up, Rob.
Yeah, and I want to say that too. I really appreciate when I know, if you're working, you're doing your thing, it's sort of an extra effort to stop and say, oh, here's a misspelling or something is wrong. But to encourage you to do that, I am try to respond within a day if people do things like that, because I just value it so much. It's really important. I value your time, that you took the time to see there was some kind of goofy thing and let me know. And yeah, so please do. It's really important to making this resource better. I want to read Sarah's iPhone.
Yeah, go ahead, Sarah.
She says it'd be useful if the genera page was separated by major classification rather than alphabetically. I'm still learning about diatoms, and sometimes I have no idea to start, except for knowing that I'm looking at an A-raphid, a symmetrical biraphid, and it would make it easier to have somewhere to start looking by having the genera separated by major morphology. Thank you for that comment. I'm gonna dig in there and think about that. How you can go to the by morphology and say get the genera that are A-raphids. Have you seen that part? Or are you talking
someone
different?
responded to my message and told me about that. I didn't know that was a possibility. So I will do that from now on.
Oh,
I'm
okay.
sorry.
Okay. Well, yeah, let me know if that is if it addresses what you were trying to get at. Thanks for the comment.
Oh, I like that. Sounds like we got a little bit of a break here as we come to the top of the hour. I wanted to share one crazy way that I used DONA one time. Actually, it was a crazy way to create content for DONA. And I only have done it one time, but we did a little event at our museum where we sat down with our high-end microscope and we had kids taking pictures of diatoms. And we gave them a picture and we said, look on the, told them how to use the microscope and had them look around on the slide, and they had to find a specimen of a diatom that we needed to put up on DONA, and they did it. And we, together with them, we did measurements, and we did some and then we, took the content that we had worked on during the course of that day, and within about 2 days, we had species pages that were developed by kids, or at least, supported by kids. And it
So
was
cool.
really one of the funnest things that I ever did with, in sort of applying DONA in a fun, Saturday morning situation. And it also made, we got to introduce everyone to this incredible website called DONA.
That's really cool.
How do we get you down there full time on the floor, Mark?
Yeah, right.
This has been great. It's
Yeah.
been awesome to have so much comments. Yeah, Layla, please let us know your thoughts.
Oh, hello. I wanted to say, do you hear me?
Yep, we hear you.
Yes.
Oh, yeah. Okay. So I wanted just to say that that this website is really very helpful to everybody, mainly as well for teaching, for taxonomy, and since you have started the marine one, and now you can sort only by all inland and marine, and you don't have too many marine. Maybe it's worth that if we can have a photo of marine species, mainly MEB species. Will you be interested to add them? For instance, for us, we are working on Arctic diatom. So probably some of them might be in the littoral of America. So, but we have only MEB. We work only with because I show the student just with the electron microscope. So I don't have the light microscope ones. But since in this we don't have enough MEB, maybe if you are interested, and as well,
Yeah, that's awesome, Layla. That's kind of it's both a dream and terrifying to me. But I'm going to defer to Tom because I'm barely learning marine groups. But
yes, please, please, submit. And if any of us can help with that submission, yeah, of course we will. Yeah, please do. Thank you, Layla.
Yeah,
You're welcome. And as well, unfortunately, I don't have a lab and money, but maybe we can, as for the International Diatom Society, we can as well give an adhesion, logging to some who want to. For me, it's worth to give each year as for the International Diatom meeting, and we are a lot of people, so maybe it helps. I don't know. Anyhow, thanks for this initiative. And
Layla, thank you for attending. You're one of our regulars to be here, and I've learned a lot from you, and that's what I like about this whole resource.
I saw you the first time in the Quebec, and it was 2016, and it was only a project. You started to think about how to make your packages, your system, statistical, etc. And so it's wonderful the work that is made. So thanks a lot. Thank you.
I'm going to give a shout out Tim Gasparic, who is the brains, the designer behind making this thing work. He just had to leave, but he was here for most of this. And I just, I'll follow up with him, but Tim is really a incredibly talented designer who came to me in Boulder and basically helped us get funding for this, for what diatoms.org is now. And yeah, it basically contributed a huge amount of his time to creating this resource. So he doesn't get a lot of credit, and he should.
Bravo. Thanks.
A really nice website. It really is.
Well, thank you, everyone. And thank you, Sarah, for joining us from a long ways away for just a great discussion about, some of our pathways forward with diatoms.org. It's just so good to hear how folks are using it and to see everyone tuning in to offer their opinions. We did again wanted
Tom, for your offer of matching up to $500. So if people can, we'll be like public radio here. Act now so we can take advantage of this challenge grant. That's a great offer, Tom, and I really appreciate you doing that.
I appreciate it. I added that please everyone go in and take this survey that that's been set up for sort of getting your opinions of DONA, as well as what how your ideas on how we might be able to make this an even bigger and better resource out there. So, all right, I think that brings us past the top of the hour. So great to see everyone. I hope that you all have a great rest of your day, and thanks again, Sarah.
Thanks,
and team.
everybody.
All right.
Bye-bye.
Bye.